So today we're going to talk about nucleotides. We're going to decipher this structure here, which consists of three main parts. So a nucleotide is a monomer. And monomer, that means the building blocks. If you haven't watched the video to monomers and the 4 biological molecules, make sure to watch that first. So monomers are building blocks. That means that they can be put together to make larger structures. Nucleotides are the monomers of nucleic acids. So if they get put together, they make nucleic acids. Nucleic acids are, for example, DNA and RNA. So one nucleotide, this is a diagram of one nucleotide. And one nucleotide consists of 3 parts. I'm going to use 3 different colors for each of the parts. So the first one is this upper part here, in blue, and that is, call that number 1, the nitrogenous base. The second part is in the middle that you see here. And this looks like a pentagon, and it is a sugar, so we'll call this pentose sugar. And the last part is the small phosphate group, which is indicated here. So that's number 3, phosphate group. Now we'll get into more detail about each of these parts. So starting off with the nitrogenous base. Put that again, number 1. Nitrogenous base. So there are 4 different types of nitrogenous bases in DNA, and there's one extra one in RNA, so there's 4 in each, and a total of 5. Okay, I'll write this down to make this a bit clearer. So the 5 different ones are A, G, C, U, and T. And these all stand for the names adenine, guanine, cytosine, and uracil. And last one is thymine. And what's important is that the last 2, so uracil and thymine, uracil is only found in RNA, and thymine is only found in DNA. So whenever there's a thymine in DNA, it would be uracil in RNA. And these 5 different types of nitrogenous bases can be categorized into 2 different types. So the first 2 are called purines. Purines means that they have 2 carbon rings. Put 2C for 2 carbon rings, and the rest of the 3, so cytosine, uracil, and thymine, are called pyrimidines. And these only have 1 carbon ring. You can memorize this by looking at these first letters. So the cut, C-U-T, cut, nitrogenous bases are the pyrimidines. So the basic structure of these 2 is, for the purines, it's 2 carbon rings. So the basic structure looks like this. So this is 1 carbon ring, and then there's another. So there's a 6 carbon ring and a 5 carbon ring. Then we've got the nitrogens, few double bonds, like this. So that's the basic structure. There's 2 carbon rings. What's different between adenine and guanine is what's added to this ring. So let's take a different color. We'll take purple for adenine. So adenine would have one amine group here, NH2. And if it was guanine, we've got the same basic structure, but instead two different additions. So we've got an oxygen here and an amine group here. That's in red. And for the pyrimidines, it's similar. So they also have the basic structure, but only with one ring, only with the 6-carbon ring. So the basic structure looks like this. And then for each of these, they'll have different additions again. So start with cytosine. Cytosine has, like the adenine, amine group up here. Then for uracil, I'll take another different color, orange maybe. So again, the basic structure, and then for uracil, it is double bond O, like that. And the last one is thymine. And thymine actually also has that double bond, but we'll give it another turquoise. We haven't had that. So it also has this double bond that uracil also has, but in addition to that, has another one here. Oh, and we forgot one thing. All of them have this double bond O here, which is part of the basic structure. So yeah, that to the nitrogenous bases. These bases then form base pairs. So when they pair together in the structure of DNA or in DNA, they always pair with the same ones. So I'll just use the shortened version. A always pairs with T, and G always pairs with C. Let's get rid of these dots. And these bond together with hydrogen bonds. So A and T bond together with 2 hydrogen bonds, and G and C with 3 hydrogen bonds. But this is then when 2 nucleotides bond together. So the second part that we had was pentose sugars. So number 2 is the pentose sugars. Again, there's different types of these. There's not 5 this time, but there's 2 different types. So we've got number 1 is called deoxyribose, and the second one is called ribose. As you might imagine, this one starts with a D, this one starts with an R. Deoxyribose is present in DNA, and ribose is RNA. The structure of these two looks like this, as you've seen at the top. H group. And the way we count carbons is always from this carbon. So this is 1, 2, 3, 4, 5. And the difference between deoxyribose and ribose is that deoxyribose, because it has the deoxy, it's with 1 less oxygen. So ribose has another OH group here. And now you can see at the top, we had this diagram here, pentose sugars here, and now you must be able to identify which one of the two it is. I'll give you a moment to think. And that one is deoxyribose. Okay, now coming to the third part, that was the phosphate group. And the phosphate group is often represented like this, in short, and the formula would be with the phosphorus in the middle and some oxygens, like this, and then it's bonded to the pentose sugar. And now what's important is also the bonds between the three different groups. So we'll take dark purple again, indicate them on the diagram here. So between the phosphate group and the pentose sugar, we've got a bond that's called a phosphodiester bond. And you can see the diester bond here with the oxygen. And the second bond is this bond here. So between the nitrogenous base and the pentose sugar. And this bond is called a glycosidic bond. Or also an N-glycosidic bond because of the nitrogen. And both of these bonds are covalent bonds. Covalent bonds. So they're relatively strong bonds between these three elements in a nucleotide. And on the other hand, the hydrogen bonds here are weaker, which is between different nucleotides. So yeah, this is an overview of the nucleotides, and the next video will be about how the nucleotides, the building blocks, combine together to make the nucleic acids, so DNA and RNA.